天早上呢，就是要跟大家讲，就是从连续昨天早上那个 discussion phone，大家就是昨天我们有一个很精彩的 discussion phone， 从每个有一些大家公司有创业的那些前辈。那些前辈来分享他们就是本身的实务经验，怎么去创立那个公司，然后在他们打拼的过程中，他们发现哪些是对来说是最重要的，去发展一个药物，然后同时呢，就是怎么去开始一个公司，然后延续公司的生命，所以就是当初我们昨天讲到说一个选题的问题。就是说怎么去做选题，那这个我是觉得说在台湾一般的学术界跟一般的早期发展的公司的大家的在经验上比较欠缺的地方。那因为我们主要是因为台湾过去20-30年在台湾没有研发的机会，然后所以我们根本是没有研发的经验，所以相对来说我们在这方面就比人家弱很多。今天我们要讲的是 Clinical Development Strategy， 所以我们很多公司来台湾做 Clinical Trials， 可是问题是我们做的 Trial 都是人家已经设计好的，所以你从来就没有学过那一段，你怎么去设计那个实验，你怎么去开始，你怎么去设立 strategy， 甚至你要去跟人家做 BD， 你说要出去要去找钱，你知道你要怎么去设定位。所以我今天第一场就是讲说 clinical development， 我今天的最后一场我会讲 business development regulatory the project management。可是第一点我要跟大家讲的是你的定位问题。OK， 好，今天的这一场的时间比较匆促一点，因为就是有很多就是我们今天我今天是一个联合的就是联合的演讲。那基本上就是说，我们昨天就讲说。制药是一个团队合作，而不是单打独斗。那我们今天在这一场也是要让大家看一下，就是说什么叫做团队合作。所以我今天有 Ken， 我有舒月，他们如果说在同一家公司的话，他们两个是我一个很重要的 team。OK， 所以 disclaimer and discussion， 今天的 discussion topic， 所以今天的内容真的是。很紧凑，然后这些都是很重要的问题去问你自己，大家自己问一下。就是说，第一个，我们常常就是说，我有一个 candidate 了，好，我现在准备摩拳擦掌，准备开始了。然后就想说，然后刚刚很多团队谈的结果，他们就说，我们就已经设定好了，我们要走这个方向去，从这个 indication 开始，因为哪家公司已经发展了这个，已经被 approved。所以我这个真的是往这就跟着他们脚步走就可以了。那我就问他们一个问题啦，说人家已经上市了，那你现在还在刚开始，那你要知道研发是一个过程就是8年到15年，15年你还有没有那个市场去跟人家竞争？因为这15年中间还有新药出来，那你15年要准备上市的话，你还在用15年前那个标准去跟人家打拼，你是不是有机会？所以就说你在选题的过程中，不是说哪家公司有什么上市了，然后你就说我就往那边走，因为那个很明确，那个脚步很明确，那不是你选题的理由。OK， 再来 build update 你的 TPP， 那就是说从2015年就一直跟大家讲，就是说你的 TPP 是你的地图。所以就说你要嫌说他太早去做 TPP， 很多东西你不知道，没有关系，因为你就是在制药的过程中，你继续向前走，你的资讯你对于你的那个 baby， 你那个 molecule 的资料会越来越多，你会有越来越多 data， 所以你的 TPP 会从原来有点不太明确，一直变得越来越明确，到最后呢，就是输液的部分。就是 labeling， 所以我今天就是第一个部分今天的第一个部分就要讲说 TPP， 然后怎么去 link 到输液的 target labeling， 再来然后从 TPP， 我们有 TPP 之后，然后我们就走到说我们怎么去选题。 我今天会告诉大家说，我们在一般的制药公司，在一些很先进的制药公司，我们在世界上很有名的制药公司，我们是怎么选题的？哪些我们会看？然后我们是经过很久的讨论，一个团队的讨论，我们最后才选了。我们有一个选择第一个 indication， 不是随便讨论的，不是随便选的。因为你随便选一个，你的第一个 indication 决定了你那个孩子将来的幸福。如果随便把它选一个，然后嫁错人，就完蛋了。OK。然后再来就是再来就是接续从那边选题之后，接续到你怎么去设计一个 clinical study design。然后我们会讲 ，Ken 会跟我们讲说，这根据他的经验，他跟 clinical 一起 work， 在 develop protocol 的过程中，哪些 study design， 哪些观念的比较先进的 study design 已经被现在目前是比较热门的。然后就是说，以前那传统式的 study design， 现在一般还是有人做，可以看那 indication 的问题。那就是说，我们一般来说，我们常会问的说 ，How can I accelerate？OK， 所以今天就是这个 flow 就这个样子。然后最后我们会做总结，然后我会 open 大概给大家10到20分钟的时间可以 Q&A， 好不好？好 ，So you select the candidate. So what?Candidate selection criteria， 所以 candidate 它是一个 compound。所以我昨天有一些同学也问我说，那你选择那个 candidate， 那你将来你怎么知道说它够不够好？那我就问他说，你是哪个 class 的 drug？What's the current standard care？ 那一样就是当初我在讲说 TPP 的话，你要怎么跟人家比？你要跟人家比是现在市面上的药，它是你 What's the standard care？而且我上次讲 TPP 的时候，同时又讲说 What's your competitor？ 哪些 competitor？ 如果说你是 novel class of drug， 有哪些目前那 molecule 是 the only molecule in the target and or you are you are novel target 一个新的 target， 可是你已经在市面，就是说在其他公司你也知道说，除了你之外，你有那个 molecule target 那个是针对于那个 target， 那你可能说其他小公司，或者是他们有大公司，它开始也有那个也有一个 molecule， 可是他们也是跟你一样，都是在第一期或是 preclinical 阶段。那你就要看说他们目前的进展是什么样子，因为你到时候是要跟人家竞争。那今天另外一个观念我要跟大家讲的是 ，first in class 是真的是那么好吗？有很多人就说啊，我这 first in class， 哇，这是铁票，那我就坐在那边等，我就坐在那边慢慢找钱，然后大家开始慢慢做。我说那你就完蛋了，因为 first in class 不是那么重要 ，first in class 给你一些 IP 的一些market exclusivity 的优势，一些 patent 的优势。可是呢，将来事实上比较重要的是 ，Are you the first into the market, or are you the second to the market？ 因为昨天也是讲说，因为台湾比较没有经验，所以我们基本上我们不会 expect we are the first to the market, but we want to be 2 months after the first to the market, or 6 months after first to the market to be the second to the market。因为那时候 market 才刚开始，所以有第一个人帮你打前锋，然后就是借着他们，就是等于是说就是真的跟着他们的风，然后你去开创你的那个市场 ，OK？ 所以这个我讲过很多次，我就是不用再仔细讲，就是说大家都知道说，你一个 candidate， 你一定要跟你的 standard care 比，或是你的 competitor 比，然后你一定要确定说，你现在的 data 就是。You had to be better in certain way。如果说是跟现在时间一样，你要知道说你比人家晚了10年 ，OK？ 因为你从现在才是 preclinical 的 candidate 而已，所以你就那个账量算，大概就是8年到10年。所以你要10年，就是说你要怎么确定说你这个会比较好？你有比较好的 affinity， 你有好的 toxicity profile， 比较好的 toxicity profile。你必须要比人家好，你才能说这是一个 candidate， 不是说你有 data 算了就算 candidate。 所以这是一个，也是另外大家有一个，就说你不是说找到一个 candidate， 然后跟人家比起来，就说好像好一点，好到那一点好到什么地方 ，OK， 然后还有你的机会是什么地方，所以你大家要看一下 candidate 的定义。Principle 这是 the preclinical stage development strategy set up， 所以呢，就在过程中，这是你拿到一个 candidate。然后你觉得说你的 efficacy， 你的 binding affinity 比较好，你的 selectivity 比较好，那个 toxicity profile 非常 clean。然后你觉得说你有比较好的 formulation， 然后你有比较长的 half life， 就说你有那些优势，我就说 OK， 我可以开始用这个开始去 develop 这个 molecule， 这才有 develop 的价值，我们叫做 developability。OK，review your science evidence。然后 ask yourself, what is your disease hypothesis if it's a novel target? What is your candidate mechanism of action? 这两个事实上是一样的问题，常常是一样的问题。Can you define your target populations? 那就是说你有几个 indication 跟这个 target 有关系？有哪几个 disease 跟这个 indication 有关系？然后所以你可以那几个就是你的 first indication 的 candidates。然后你从那边去分析 ，build a TPP for each target indications, craft a future drug labels， 然后 choose your first indications, and then you continue the good development foresight. So foresight. So create a set of the scientific data to support your support your hypothesis. 因为这是 building 你的 IND package，OK。所以 IND package， 你把你的想法放到纸上，那个大家都会做。可问题是，你那个 package 看起来够不够 smart， 那是另外一件事情。所以我们通常等一下也会跟大家介绍说，我怎么去 build 一个 package。那等一下就是在下午第一场那个 regulatory project management 的那个 section， 我会提到说我们怎么去 build 那个 story。OK。So update your TPP。Regular 昨天我讲过，在一个 section 就是出院的那一场有说过有3种 TPP， 一个 TPP 是叫 regulatory TPP， 那这个 TPP 呢是 FDA 它有一个 guideline， 它有 suggest， 它是 suggesting， 它不是说 you must have，but I will tell you say if you have that。It will be much easier to communicate with us. OK， quality TPP， 然后 QTPP Frank 2年前有介绍过。那也就是说，然后那些 TPP 就是说那些 criteria need to be met in the product development process， 也就是 CMC 的 process。它 dose administration， 它的 formulation， 它的 solubility。那些就是它的 quality 的 particulate number， 那些对它的 specifications need to meet. We will set up the TPP for that, and then we need to meet. When they develop the product in the CMC section, in the CMC part, we need to meet that. And then we have commercial TPP. So commercial TPP involvement in the beginning is not too much. The most part is we use commercial TPP as a standard to guide how we're going to choose the first indication. OK， 所以可是 commercial TPP 它基本上就是很小，一直到晚期的时候，这个 commercial TPP become much more clear， 因为那是要准备 commercialization、market launch。OK， 所以这个就是 TPP， 然后我这样子。这是一些 criteria， 我们会看到说，这是 regulatory TPP 的部分，不太多，因为早期你对你这个 molecule 还没有那么多了解，所以这主要就是在 drug administration、drug 的the strengths and forms 的 the supply storage handling，OK， 然后这个部分呢，从这里到这里。这是什么 ？Quality TPP， 因为那时候你要开始进 manufacturing， 你要可不可以 scale up？ 它进去你要去怎么测试，就是 synthesize 之后你怎么 purify 之后你怎么去 test， 你要 test 完都 meet the specification 你才可以 release， 所以那时候这个是很明确的 ，OK， 它跟 chemistry 有关，不管是 large molecule 或 small molecule 一样，就是它有 certain specification you need to meet， and commercial TP is very little。And you can see very little information you will know in the commercial TPP. The shaded area 是有资料的，可是问题是你会发现它资料都很少，因为东西你知道，你对这个 commercial 的要求并不高，那最主要是在选题上面。Update 有 TPP， 这边是这是2年前的那个 TPP 的那个 symposium 的资料。那基本上就是说大家有兴趣的话，可以回去网站去找，我们都有录影，所以可以听一下。就是说，它主要是在 end of preclinical stage、end of phase 1、end of phase 2A、end of phase 2B 或者是的 phase 3，然后每个阶段在你开始要进入这个 preclinical 之前，所以你刚要开始之前。你要 update 你的 TPP， 你要知道说 what's your target， 记得这是你的地图，所以你要知道说你要 meet what's your standard， 你的 how high is your bar， 然后你结束之后再要去 present 给你的 senior management， 跟他们讲说 I finished my preclinical development stage， I'm ready to submit IND， my IND package is all ready， I need the budget support for next stage。Of phase 1， because when senior management review， 我昨天有讲在 portfolio management 部分，就是说 senior 在 milestone review 2件事情，一个是 all your data to tell them to declare， 去跟他们讲说你完成这个阶段的发展，因为你所有的 checkbox 在这个部分你的 data 都完成了，你有一个很好的故事 ，can tell your senior management say I am ready to submit IND, start a clinical stage。这是一个，另外一个就是 clinical study plan, phase 1 study plan， 就是下一个阶段的 plan。然后另外一个下一个 study plan 包括什么？包括 FTE， 包括就是说在这个阶段，如果是要进入这个阶段，我有哪些部门必须要加入我的 team， 去一起开始做 phase 1。还有，我要开始做 phase 1， 在每个 function 我要多少钱，那些我都要做报表要做好。所以我进去我一个 slide， 我进去做 presentation to senior management， 我20 slides。The 10 slides is what kind of data I have to support if I say I finish my preclinical stage development. Each function only had 1 slide. It's not a data show. It's not data dump. I just finished that last Wednesday, and so next 10 slides, the 8 slides is the clinical study design proposal. 最后2个 slide 是我的 slide， 我的 slide 是在这一年，如果说开始做 Phase 1A single dose escalation。我需要哪些 function 来我的 team？ 另外一个 slide， 那是一个部分的 slide， 然后那些人的薪水要多少钱？然后另外一部分，但是我们叫 FTE， 另外一部分在同样一个 slide， 我另外一个 line cost all the pocket cost， 那我们就说 oops， 在我们的 organization， 那基本上就是说，它那个 cost 就是说，在那所有的 function。在做那些东西，每个 quarter 我要多少钱？好像说我有 CMO 有 CRO， 那些所有的钱加起来，每个 quarter 他们 out of pocket how much？ 也就是说 senior management 当他们，所以我就是说这是一个部分，就是2个 slides。然后基本上他们 approve 的话是说，他们看那个 data， 他们问一些问题。然后第2个 slide 就是看我们的 clinical study design， 他们会给一些建议，然后我们会说好，我们就回去再转。可是他们最后会 approve 的话 ，approve 是看他们的 approve 是他们不是随便 approve， 他们会跟我们讲说，哎，你这个 design make sense， 然后他 approve， 同时就是 approve 我那2张 slide， 我2张 slide 多少FTE， 我可以拿到多少 FTE， 那2张 slide 告诉我他可以给我多少钱，所以我今年要了13.2 million for the phase 1。For the preclinical stage， 因为最近我是做那个 preclinical stage 的那个 presentation 的 request，so I asked the preclinical stage I asked 13.2 million, and the first study the single dose dose escalation study， 我要6 million， 所以就是说他们 approve。那个 price tag 同时也是在一起，所以他们不是随便 approve， 因为他们知道说他们 approve 多少钱是 involved，OK， 所以大家就要看一下说，所以这个不是随便写，就是说随便想说你过去跟人家要钱，你要跟人家讲说你要多少钱，而是为什么 ，OK， 所以现在我就是也开场，然后让大家慢慢想一想，然后我这样就 pass over to 苏月。
我的 function 是regulatory labeling， 过去10几年，我的 background 是 pharmacist， 我是药师。那在美国，我做了10几年药师，然后又做了10几年在药厂。那我一直在 regulatory labeling。那我想先解释一下 labeling， 就先不要想说target labeling， 解释一下，就是说product information， 比如说那个你们在那个药一罐拿出来，然后里面不是一张访单吗？那一张访单通常你们拿出来就丢到垃圾桶里面去，看都不看，对不对？那你可能不了解那个上面写的 information， 就是刚刚类讲的花了$30 million， 你知道吗？这个 whole product development 最终就是 what's written on 这个访单上面的。那个 health authority 会 approve， 那决定说你这个药可以用在什么病症，可以用在什么样的病人，可以有什么疗效，那有什么那个注意那个要注意的地方，所以这些 information 都是整个这个 product development 做了很多的那个clinical trial， 那最后的那个 data to support 这个 labeling， 所以我想解释一下这个 labeling 是什么意思。那我接下来就是类刚刚解释，那我跟你讲，这个其实他 pass on 这个 microphone to me， 其实是 real life situation。那真的是 real life situation。我常常那个不久以前也是一样，我会收到 email 或者是 phone call from project manager like Lei，OK， 他会跟我说 ，So we are about to, go to FDA, okay, for a meeting with FDA. Or 他会说 well, we are thinking about phase 3 studies now. So I need you to do a target labeling. So this is a real life situation, and I'm going to take you through what I do when I get phone calls from Lei, okay? Okay, so explain to you what is target labeling. So target labeling, we explained to the labeling is the product information. So target, now you add the word target, is expirational labeling concept. Because we said labeling is the result of the study, but at this stage, we have not done any study. So there is no data to support anything. So if we are going to write a label, it's aspirational. Right? It's your dream. Your dream about what you would like to be written on the piece of paper. Okay? So it's a concept. It's aspirational. And that's what the sponsor, which is a manufacturer, intend to market the product with. Okay? So that's a target labeling. Okay? So the goal is, at this moment to think about this target labeling is, I want to know, as a company, what I like, what would I like at the end? I want, what do I want it to look like? What would I want it to be written in this product labeling? So then you can plan your product development with that, that thinking in mind, okay? And so it's almost like you're writing the final products, in the earlier stage. And so this document is a living document because you can do this actually through different stages of your product development. It doesn't have to be right before the phase 3. It can be before phase 2 study. Anytime you are thinking about developing a study and then you have question, you can always do this exercise and I'll take you through. Okay? Okay, so the purpose, to me, two main purpose. One, just like we talked about, to guide the design of your study and how you conduct the study and how you analyze the study. Remember, when you present data to the health authority, it's not any data that you come up with. You have to pre-specify how you're going to analyze the data. And then the agency needs to agree with your plan on how to analyze the data. So by discussing this, the target labeling, you will know I need to analyze the data a certain way in order to achieve the result, the data to support your labeling text. Okay, so that's the goal number one. And the second purpose is to use it to help you, guide you, to support you with the discussion with the health authority, and I'll show you how you can do that. Okay, so this is a flow chart of the product development. As you can see, phase 1, first in human study, proof of concept study, and then they just mentioned about, right, you will select, the molecule, you start to decide, okay, if I'm going to take it to different stages of the product development. So, with the gold star for target labeling, you can do that, in different stages. And this is just to illustrate to you, you can do that. And sometimes I do have that, before the dose ranging study. You want to know how many doses should I test with. And this could be an opportunity. You can do target labeling discussions to help you decide on that. And most cases are before phase 3 study because that's the most important study to for the registration. Okay. And this is for the second purpose. The target labeling, use it for your, to facilitate your interactions with the health authority. So I have two examples, US and then EU. So with FDA, typically, at the end of phase 2, you will have a meeting with FDA. Actually, yesterday after the afternoon session, a lady came and asked me about this, question specifically for that because she's about to go to FDA to have a meeting with FDA to discuss, the possibility of registering the products in the U.S. So, and they already have a phase 3 study. So, she was asking, how she can do about it. This is, it's here. This is what you can do to, to do the target labeling, and then because you're going to have questions to the FDA, you're going to ask. I think the one question you will have is, I intend to have these indications for these products. Does my study support, this indication? That's always the number one question you have in your briefing book to FDA. So by showing, the indication language, exactly what you intend to put in your labeling at the end of, the development, that can show FDA, okay, this is the exact language you would like to have, and they can evaluate if your study can support it or not. And that's just one example. You can, put in your, clinical study sections, showing that you would like to claim, the product significantly reduce certain things by percentage, that kind of language can help FDA to understand exactly what you intend to achieve with the phase 3 study, okay? And also, I think right before the submission, you might meet with FDA, and potentially you can do the target labeling and then present to FDA. And in EU as well, oftentimes you have scientific, meetings with the EMA, CHMP, and you can include the target labeling information in the briefing book. Okay, so importantly, how do you prepare? So when I get phone calls from Lei, then I need to figure out how I'm going to prepare the target labeling. So first I need to know what are the competitors' products. And Lei mentioned that as well, right? We need to know the competitive landscape, what are the other products on the market that could be our competitors. And if you're first in class, it's going to be challenging. It's really challenging, and you probably will look for something that's similar or maybe by mechanism of actions. So you need to do some research to look for that, and then look at their labeling text, and that could be your guiding document. And you probably want to understand if there is any class labeling. I think like diabetes drug, they have a class indication. So there is no you don't have to be creative. It's just same language for everybody. And also understand the agency's guidelines, regulations, or their expectations. So we'll guide a team that realistically, this could be the labeling language that we will end up with. Okay, and then when I do the target labeling, I like to do scenarios. And because I don't just, it shouldn't be just one options. There could be a base case scenarios and something that fallback means if I don't get this, what else can I get? And of course, you want to go with the best language that you want, but then also you want to think about potential. I may not get this one, so what would be the next options that that might happen? Okay. And also another important things, I need a commercial TPP, okay? Commercial TPP actually drives the target labeling, because I need to understand what a commercial position the products, for eventually when they launch the products, how do they, vision this product will survive, I guess, in the market field, okay? And if hopefully there's already some preclinical data or some limited clinical data, if they are available, then it will be helpful. And this is a template that actually I developed, but it's very similar to the FDA's TPP format, but it has Okay, so it has all the this is the commercial TPP information, and this is the target labeling text that you can put in, and then study design to support. And then I have a column for regulatory assessment. Sometimes they will give us some comments. We can document it, and also my own comments. And this is internal documents. This is not to be shared with the health authority. For communications with the FDA has a specific TPP template. Then usually I will lift the information from here, put in that template, and then submit to FDA. Okay, so this is just for your internal use. And I just made up this this example just to show you how you can do it. This is the commercial. Let's say this is the commercial, TPP, and they would like to say that it's indicated for reducing the signs and symptoms for in patients with disease A. So then I will discuss with the team and then come up with the scenarios, different scenarios, because do you really want to just say reduce the signs and symptoms, or you want to say it's for treatment? Okay, so they are different. And you do need a different study design endpoints to support different indications. And here I added, well, do you want to go into adult or pediatric patients? That's a good question too. What kind of patient populations? And by knowing exactly what you would like in your indications to be competitive in the market, it can help you understand, how you can design your phase 3 to achieve what you like. And also, I like to do US versus EU as well, because they are slightly different. In the EU, it's very specific. Each indication you have to specify if it's for adult or pediatric, an age group. So it's it's good to understand the EU potential language to help you. Okay. So the considerations when we are working on target labeling, this is done not just by me. It cannot just be written by myself. I have to take it through cross-functional team discussions. They are, I guess, representatives from clinical, medical, safety, commercial, regulatory. It depends, if I have preclinical data to, PK, right? Really depends on the topics you want to develop. And it's important you set the goal and timeline so you don't drag, for a long time, so you can achieve. And you need a full commitment from the team to, to finish this target labeling. And then more importantly is you need the management support. Okay, you don't want to just develop this and then it just goes nowhere. And usually I like to, because I think Lei said, she's a helper. She will present, the clinical trial protocols or the plan to the management. And this would be good timing, to present this information, to show the management, guess what? With this design, this is what the team thinks, the target labeling is going to look like. Okay? All right. So I'll pass it back on to Lei. Thank you.
So one thing I want to say, also the team, there's one really important function is biostatisticians. Because trying to set the bar, although the target labeling is an aspiration, your expectation, but you need to, as time goes by, you need to align with reality. So you can say, best case scenario, I want my bar this high. Over time, when you have finished a phase 1 study, is that bar you still can meet the bar this high. And then you go to phase 2, is that bar still this high? And phase 3, is the bar still this high? And then is the label, the language, do I need to make an adjustment? And so biostatisticians, they will based on the study result, they will do the analysis. They will tell us, can we claim this language, can we use this language to say our efficacy look like this? So that's how very important the biostatistician, sometimes we didn't really think about, they're really important. We think they're just playing the number. But actually, they are really important. Okay. So next one, we are going to dive into the clinical development strategy. So the first thing we want to talk about is how to pick the first indication. It's very, very important. It's just like marriage. Okay. So you pick the wrong person. Yeah, you know what is going to happen. Yeah. And in the reality, you can just divorce. That's fine. But for a drug, it could mean just like the drug just died. So that's very different. It's life and death, the situation. Okay. So assessment for the indication decision. Do you have enough scientific evidence to support the decision to choose one indication over the other? Because that's science first, early development. I'm not saying, your instinction, just like trust your instinct. Just you have one molecule, which indication is the best? As a scientist, your gut feeling. But gut feeling is not telling you. Gut feeling only going so far. If you just continue going by your gut feeling, you are going to have problem. So next question is, does regulatory and business environment support your indication you choose? Okay, whether you get that. Okay, so as a scientist, I know in Taiwan, we has been past couple years, just while we were, that's how we do science. In the tipping scale, in the scale of development, drug development is tipped in this way. And because we don't really know how to evaluate the environment, we don't, what kind of environment we are looking at, we don't know. Okay, this year, and I want to, using this symposium, to tell you, we need to tip you our balance back. Not only look at scientific environment, everybody are very good at that, but we want to look at environmental evidence, whether that support my development. What is your business environment evidence? Here's a checklist. What we usually do in the pharmaceutical companies. What's a medical need? And I think recently everybody really good at that too. But I do want to say something about that too. Everybody loved orphan drug. Everybody loved orphan drug. And then a lot of time I'm looking at people's proposal, they will say, I have opportunity, two things. I'm the first in class. So I'm guaranteed I got a ticket to the success. Second thing is telling me I am going for the orphan drug indications. Okay, I'm going to tell you why I don't want. That's a good idea to have, but not necessarily give you the ticket to the success, okay? Current treatment paradigm and their outcome evidence evaluation criteria. What's the treatment paradigm? It's current available treatment options. 也就是说 what's the standard care? What's the available drug for the treatment for particular disease? What kind of patient population? It's something we need to know about is currently a lot of treatment, the drug treatment paradigm has become so fragmental. 拿一个例子来说，等一下我会给你一个例子 ，non-small cell lung cancer. Everybody knows that. Everybody loves it. But that's scary. Third one, the same thing. It really is linked with that too. Competitor landscaping, market size, market differentiation. Which one is more important than the other? That how to make decision. How do you cut into the market? 怎么选题？昨天讲的。How to pick a topic, how to pick the first indication. And then the recent trend is patient voice, patient's voice. Is the current standard care or current treatment paradigm enough? A patient wants more. And that's your opportunity. That's why you want to look at that. And then next one is patent strategy. Do you have not patent strategy? Not just apply for the composition patent. Something I have seen over the year, or the method of use. I think it's very good to see everybody know how to file the patent and know to file the patent to protect yourself before you go out to talk to the other people. But do you have really a strategy there? I have seen some project team, they file 10 different composition patents. Or they file a lot of patents in different countries. And then will that give you enough protection in the direction you want to go? After you spend so much money, okay? Regulatory pathway and a strategy. Whether that environmental regulatory strategy is work, it really support your choice, okay? Again, why we do that? To the end, we're talking about the death. I would say death trap, or whatever. And but really is trying to see, here is a development cost. Whether to the end I can have revenue, 我可以赚钱赚回来 to overcome, to make it even, the development cost 赚回来。同时，什么时候我可以开始有 returns of investment？ 为什么 ？Why? Because I'm sorry. Because you need to answer to your investors. Remember yesterday, James, he's going through funding A, B, C, D, E. It sounds really great accomplishment. But if you ask him, what made him wake up in the middle of the night? Is whether he can fulfill all the contract he signed to those investors for the certain milestone payments, whether he can pay that. And he had to figure out the way to get a return. Okay. So that's why I don't want to be a boss. So I don't have to look for money. Good to be a project manager. So a medical need. So what we are looking at, here's an example. So current treatment, say treatment option for rheumatoid arthritis. Everybody know that. It's very hot topic. Very crowded environment. You can see how many drugs are available here. I would say if we count individually, more than 15. It's very crowded. Can you get in? Is it worth to get in? That's a good question. Non-small lung cancer, everybody loves it. Here's a picture that you see how crowded how fragmental this market is. Everybody know. We have science progress into the point. We know for non-small cell lung cancer, how many subtypes we have. Where is opportunity? Do we have opportunity? Don't be afraid. If you have a strategy, you know where to get in. The same thing with rheumatoid arthritis. So, here's a question. Competitor landscaping, market size, market differentiation. The point is, don't be afraid, don't be shocked by those pictures. Because if you know the science, you know the market, you know where is population, what those science stand for, why those drugs are effective to those populations, you can find a way to get your drug in. And that's the point. So in these 2 years, there's a concept called the disease interceptions. So it's not, there's nobody is going to tell you, say, okay, here is an empty spot. Welcome to come in. No. So let me go back to here. There's a drug, we call it disease intersection. It's when you have a molecule, especially a novel class of target. What we will do is we will start looking at where is the market opportunity. If I know that is specific for certain target, I may have opportunity, and those targets maybe have additive effect with certain molecule. I will go for the combination therapy. But before that, also I will look at that. If I have effect as adjuvant, to combination with the other homotherapy with, or not necessarily for this one, not for homotherapy, but the radiation therapy combined. Or I can pick up whatever failed in this class. Because if my combination therapy with this class of drug, existing drug combination, have additive effect, synergetic effect, I will say I will cut in here to do a combination because I found a synergetic effect. Give me the opportunity to make the drug combine with current therapy much better. So I have opportunity to cut into certain type of therapy. Also, I will test whether drug is effective when the drug patient, they fail this line of therapy. They fail the current ELK drug therapy, and I will pick, and then, but after they fail the ELK drug therapy, the patient still responds to my drug. I will pick up here. I will pick up from here. Whatever failed. So you need to know how to enter disease, but don't be afraid by this. but know where is your foot, where you can put your foot into the door. So from there, you evaluate your market. That's your opportunity, okay? The same thing with this one. If you put the chart like this, the same thing, ask yourself the questions. Are you going to be in the beginning of the treatment? Can you cut into the stage? Which will give you a huge opportunity because you are going to be able to touch the huge market for the compete for the naive patients. Or you will pick up whatever failed. And I can tell you in the past, so that's how if you have the same class of drug, you will see that's something you need to lay out for yourself. That's called the competitor landscaping. So how many drugs in the same class, say here is the same class drug, when they come in, you want to be second in the market. What's the sequence? What's the rank you are is going to arrive marketplace to become a drug in the same class of competitors? Do you still have opportunity? How much behind you want to be behind them? Also, you want to compete with those drugs in different classes. They are going to be approved at that time. You need to know where you are going to come in, and do you still have opportunity? And you make decision whether it's worth it for you to go for these indications. Okay. So for example, if I see it's so crowded, this drug is my competitor in the same class. There are two drugs. This drug, if I say I'm going to go to the market in 2015, I'm the fifth in the same class. I'm too late. I'm not going to pick up any market. But if I'm not the first one is going to be 2017 or 2012 to be approved. I want to be second in the market. I'm going to set my launch day in 2012, 2013. And I will ask myself, can I run that fast? If not, I cannot choose this indication. Okay. The next one, opportunity for market differentiation, the same thing. You have to be able to differentiate yourself. Even you are compete with them. You want to see how's the different classes. You want to see how can you differentiate yourself from the other class. You have to ask yourself whether you can have better safety profile, you can have better efficacy, you can have cheaper in the price, and you can be easier to use. For example, you can put your drug into an auto-injector. So patient can inject themselves, they don't have to go to a hospital. Or you can make it as a patch instead of injection, or you can make it oral instead of injection. Those questions you need to ask yourself, and that's about the formulation. So that's related to the CMC development. So that's why I'm saying the product development, CMC development is equally important because they help you to achieve your wish list, okay? And then you do the decision, the disease intersection point. Where is the treatment entry point? I have described that already. So here is how we choose, how we look at it to choose a first indication, okay? And then the risk without market differentiation. If you don't differentiate your market with your competitors, that matter is same class or the different class, market application may not be approved because you are not as good or you are not good enough in compared to your competitors. FDA is not going to approve because they don't think. One thing you need to do is a risk-benefit profile, and FDA, and especially from EU, they will want to see that to say, is your drug is better, and is not the same, and provide some benefit, even better to use, easier to use, you still can be approved. Sometimes if you are not as good, but you are easy to use, and you are cheaper, you can be approved. Okay. The other one is very important in the US, in the EU, is whether you can be accepted. You will be able. The other thing is a lot of drug actually in US Oh, they got approved. You think that's the end of game. Or you can just sell your drug. Guess what? You are not cheaper. You are not as good as the current standard of care. Nobody is going to buy it. So your benefit, the penetration of we call it market penetration, is how many doctor they're willing to change their prescription from the current standard of care to your drug. No, because they have current drug they use. They have a lot of experience with that. They feel it's good for their patient. They're not going to change their prescription for you. You are not going to sell. Okay. You won't get a reimbursement, even worse, in the US and EU because everything is by insurance company. The insurance company not going to willing to pay it. Are you going to sell it? No. So basically, to the end, you got to prove it doesn't mean anything. Okay. Patient's voice. Again, I say earlier, rheumatoid arthritis. Recently, we finished a patient survey, the market research. We asked patients, 30 patients sample size. We asked them, say, what's important for you for this drug, current drug availability? Current drug, they have go to a point, we call the ACR 20. It just means you have 20% improvement in your life. Symptom improvement, 20% improvement. And the 30 people, say 100 people, we call the ACR 20, 60%. It means 100 patients, there are 60 patients, they all experience, they use the same drug, they all experience drug. 60 of them out of 100, they feel the symptom relief by 20%. It means their life, from they wake up in the morning, they feel not as stiffness. It's still stiff, but it's not as stiff by 20%. And then they say, that's drug we have at this moment. So we ask them, say, what do you want in the drug? So they tell us, say, I want to be get up. I wish I would have a drug. When I get up in the morning, I can just get up. I don't have to wait 10 minutes, 30 minutes to get up in the morning. And I will be able to know, the RA patient, they will have, when they get flare, it's very uncomfortable. Whole body is painful, and they will, one day is wasted, and they cannot really do anything. So they will say, I wish I know which day is going to happen. So patient's voice give us no. RA drug, does we still have opportunity? Yes. But you need to know how to get in, okay? So I'm not going, because of time, I has been stretching, I need to give time for Kent. So that's how we need to know, just like what's the patient's voice, give the opportunities. Patient's pattern strategy, again, better pattern advantage. We have known about that. Later on at the next sections, Wei Lin is going to give us some discussion about the pattern strategies. So we will talk about that. It's more is better. And what kind of pattern protection you need to have. Okay, the composition pattern I talked about last year, method of use. I'm not going to go forward more with that, but leave this one for you to ponder, to think about that. And the original part, the composition pattern give you 20 years. Tracking back to the day you file the patent. Data advantage, data exclusivity based on the method of use only give you the regulatory from the day you got the regulatory approval. Okay. And the pediatric give you additional 6 months. Okay. So I'm not going to go forward too much about that. That's all the information for you to read about that. More detail I introduced last year already. You can go back to the website to look at the information. The point, again, so orphan drug, again, I want to say it's a very good to enter the market. Give you the benefit is you can get a regulatory accelerate approval, the treatment how to say, if you have any question, you call them, they will give you an answer. They will help you to do your development, give you a lot of feedback, because a medical need is market small. You will be able to get a waiver in money, say that you ask for you will be able to waive a lot of process fee, that as for FDA meeting fee, those you can get a waiver. But is that a guarantee for you to get market approval? No, because market approval, your data. If your data cannot support that, no, you are not going to get approved. So we need to know that. And then you also get some different country get a tax benefit too. So that's a good thing to have, but don't mistakenly say this is a ticket for you to the approval. So everything I talk about, how we are going to decide the first indication. Okay. Yesterday, if some people come to my section in the portfolio management, do you know the SWOT? What is SWOT? Strengths. What is the strength of your product, your drug? What's the weakness? What's opportunity? And where is opportunity? What is the threat? Put them into the chart. Put them in. Yesterday I said some of the threat is because unknown. 你不知道，它就是 you have uncertainty because you don't have the data. Over the time, 可是你要知道说 where is uncertainty 在什么地方 ？Where is uncertainty? You build a strategy to make it certain, to get the information, to make it become certain, make it from the threat become opportunity. So this is a chart. You will do it, exercise again and again with your team to see where your weakness, where is the strength. So that's telling you, give you information, which market you can get into, how can you differentiate yourself from the other current treatment paradigm, and what's your weakness, what's your strength. So how can you develop your drug into that to compete with everybody. And so you build this one. So you build a strategy for individual indication candidate. 如果说 if you have 5 indication, you think your target all touched, you will build 5 SWOT. Individual to analyze it. So you know where is your opportunity, where is your strength. Pick the one, give you a good, know your strengths you can achieve, or you have good opportunity to achieve. You can overcome some weakness. The weakness you can overcome. You know the indication, the opportunity huge in your drug. You know you can do it. You know yourself the best. You know you can do it. And you know you can put opportunity, you can put a strategy to turn the threat into opportunity. If there's a 1 indication or 2 indication, you at that time you need to play with your gut feeling. This chart help you to, so you can use your gut feeling, your 直觉, but you need to have this one as foundation to do the decision based on your gut feeling, because when this is your reality. So you are not just pointed in the dark. You're just shooting the dot on the wall. You don't know where you are going. But doing that, you know what to do. And I think, sorry. So let me go that really quickly so I can give 10 minutes to talk about the next step. You choose an indication, and you need to build your clinical development strategy. So after you choose the first indication, you need to develop a story based on your hypothesis. So you need to tell the story. So I'm saying, you are not just putting things, the data dump into the R&D package. You need to strategize how your story is going to look like. You need to tell a really good story. And then you need to you need to design the study will be able to prove your hypothesis. You need to design the study will show the data your biologic data response. You see the biologic response touching your target by treatment, so that in vitro and in vivo, before you get into a clinical study, you need to be able to predict your dose selection, be able to predict your potential issue in safety. I'm talking about the weakness. How can you turn the weakness to become opportunity too? Predict your outcome. Design a study, clinical study design. It's not just say, I put it into the formula, everybody know how to design a study, and I'll just put into dose escalation and 3 plus 3, 3, 3, 4, 6, 6. Everybody know what to do. But you want to design your clinical study knowing which should indicate which population you are looking at, what kind of endpoint you want to measure, and to answer my question. There's opportunity for you to answer the question. Based on your assessment for development environment, how can I disease intersection? Can I really cut into a disease? And that's based on my clinical studies, okay? I need to design that to answer the questions. How to design the clinical studies to prove the safety and efficacy and to the POC. And that's, once you can reach your POC, you can say you really have a drug, okay? Opportunity. So every drug has a story. Build a story using your data, okay? I think I don't have to say more. Everybody are very good in that. Design a clinical study to support your study, to support your story. New paradigm study design, patient's voice, physician's voice, market analysis voice, what they want. Not just design a study, that story, design a drug as because I like it, okay? How to design and effective the proven safety and efficacy, how to attract patients to enroll the clinical studies, how to improve the patient enrollment, how to evaluate your outcome, what kind of outcome you are going to measure, what is your TPP? Again, go back to TPP. Again, without further ado, I'm going to pass this one to Ken. Take your time.
谢谢雷。早安，你好。That's about the extent of my Mandarin I'll be using this morning. Thank you. Thanks for your patience with my English. So I work for Janssen Pharma as a medical writer. I manage all the early development medical writing across all the therapeutic areas. But I focus in on oncology, and immuno-oncology is the hot topic right now in early development. And so we're looking at creative designs in clinical studies. So traditionally, the clinical development has involved a lot of preclinical work to establish the safety of the drug and some preliminary pharmacokinetics and pharmacodynamics, and to develop a good justification for that initial dose that you're using in the first-in-human trial. Then you do a single ascending dose trial, a multiple ascending dose trial, phase 2 trial, maybe a phase 2B also, and then phase 3. So it's very stepwise, conservative approach. It's still appropriate in many cases. And, in today's competitive landscape, as Lei has described, we're looking at trying to get the drugs to market quicker, to get the drugs to patients, to help patients, survive, cancer and immunologic diseases and so forth. So we're looking at, various accelerated designs. We're doing for combination therapies. We're looking at a lot of combination therapies in immuno-oncology to see what works. We're looking at different ways of modulating the regulatory T cells, the effector T cells, and so forth. So once we know the dose we think will work in a phase 2 study, we may move in right away into a proof of concept study with combination drugs with a phase 1b safety run-in, maybe with only like 6 patients, and then trying to move to phase 2 as quickly as possible. And we may even have an interim analysis, have an end of phase 2 meeting, maybe with the FDA or EU, and amend that same protocol and turn it into a phase 3. So late development, I'll probably skip over that in the interest of time now, but basically, you know, as I said, we could even do a phase 1b/2 and phase 3 and get to that regulatory submission very quickly. So one way of doing an adaptive study design is doing this Bayesian adaptive design in early development, phase 1 studies. So it's an opportunity to make decisions earlier. Frequent safety analyses by a safety evaluation team. It's very dynamic. And it's, we include a lot of assessments of biomarkers and PK and efficacy actually in a phase 1 study for oncology because we're enrolling patients. And so we can add or drop arms if appropriate. A trial can also stop early if needed. So this adaptive design can involve an accelerated titration where we're dosing one subject and then the next subject 2 days later. So we're getting rapid information about individual doses and accelerating the doses as quickly as possible. So we're actually exposing less patients to a drug or dose that may not be safe or may be ineffective. So we're getting ideally to the point of deciding on a phase 2 dose quicker and with more benefit risk in mind. So this is a graphic on the adaptive designs. It requires a lot of active data collection. So the study sites really need to keep the database up to date and available in a dynamic way for assessment by the safety evaluation team and the physicians at the company as well. And then, so there may be an interim analysis. There's different ways of doing adaptive designs. There may be sort of a formal interim analysis plan or more of a dynamic periodic safety evaluation team meeting type of plan. So the benefits of an adaptive study design, it mitigates the risks with larger and longer trials. Could be less expensive in the long run. Improve data use and provide flexibility to identify the right dose quicker. We call it the recommended phase 2 dose, RP2D. You may see that abbreviation in clinical studies. An opportunity to stop for futility or early success if data is compelling. Again, to amend that protocol to even make it a phase 3. Or at least to the randomized phase of a phase 2 study. Again, reducing the sample size, allow earlier decision making, and it requires a lot of, as I said, a lot of analysis of the dose that you want to study first. And it doesn't mean you don't have very clear guidelines in the protocol. You may not list every dose in the protocol. You have flexible wording, but you have very careful wording of how you're evaluating the doses as patients accelerate in these early development phase 1 studies. The FDA does have a draft guidance on adaptive designs from 2010. The EMA just came out with a draft one in November too. So I think that's all I'm going to say today. I have a session next actually on protocol designs in the next session. So you're welcome to join that. Thank you very much. I'll hand it back to Lei.
Thanks. 所以我们就是时间差不多到了。那如果大家愿意的话，我们再延伸10分钟，好吗？就是我希望有一些 Q&A 的时间。然后最后，我们刚刚就讲过这些。最后一个 in conclusion， 所以 clinical development 是一个 extensive and complex team sport. I like to talk about team sport， 因为就是让大家有，就是我们要作为一个 project manager， 我的责任是 recognize everybody's talent, who can do what, and group the team together, and empower everybody to contribute your ability and work together as a team and to get things done quickly and with good quality. Continual assessment with the entire development environment is critical. We need to know, always keep it in mind. We are not working in the vacuum. We are working with huge environment, very complex. We need to know, keep eye on what this environment look like, what they need. So talent pool is in this multidisciplinary team work as a key. And that's what I come in. I work as a project manager, a global project manager. I work with everybody. And my team need to know what they stand for. They need to know they have a ability, they have the freedom, they have the freedom to speak out based on their expertise. When we work together, the good project management is critical to ensure the seamless operation. They need to know what is going to come, who is waiting. One function is waiting for the other function to finish their work, and if they don't know, this machine is going to get stopped. Risk benefit. We need to continue evaluate risk and benefit. To the end, most important thing, don't lose your focus. Follow your map. TPP. Thank you. So now is 10:20. We have the start coffee break to 10:40. So if you like, we can use 10 minutes to open for the Q&A. So with 3 of us, we are here to answer your question as well. Do we have any question? Okay.
刚刚讲到 adaptive design 啊，就是大部分我们看到是在 oncology， 有没有在非 oncology 的？就是的情况，它是大概是因为通常 phase 1都是 healthy volunteer， 那 phase 2才会进到 patients， 所以它是怎么样让它缩短，或者是用在非 oncology indication 是在哪一个部分，是 late stage 或 early stage 就可以去用这个 adaptive design。
Okay， adaptive study design 它并不会 replace the first except oncology, you are right. We all have to do the healthy volunteer study. And so adaptive study design not started from the phase 1A. Everybody had to do a phase 1A, dose escalation in the healthy volunteer, except oncology. Of course, we know that. The adaptive study design, the beauty of adaptive study design is start from after phase 1A, you finish your single dose escalations. And in the old time, we have phase 1B MAD, and we have phase 2A POC. What we did now with the lump up with phase 1B, as what Ken just say, phase 1B MAD, run in maybe 6 patients, 2 dose, 3 patients for each dose to look at the safety and efficacy based on the phase 1A SAD study data. And we make decision very quickly, and we may add more than one indication into this run-in study to do the evaluation because we want to see what disease interception point is going to be. And then we will decide very quickly which indication we are going to choose. And we we get into the phase 2A POC very quickly. But everything we design in one protocol, and we run into this cycle. So we design run in, we get in, we analyze that, stop. Whether that work, run it again, stop, and we make decision which indication we are going to. Run one cycle, the first cohort, stop. Many different stop of the interim data analysis. That's how it look. So in the beginning run in, and here's an example. This example is for the phase 2, phase 3 study actually. So each cohort we have a patient, and we run one cohort, finish running, stop, interim data analysis. And then we see the safety and efficacy, and then we continue next cohort, stop, until we see the efficacy, and evaluate the efficacy whether that's good enough, and we stop. So everything designed is in one protocols, and so there's no run in, stop, and then start another protocol, because most time wasting is when you finish one protocol, finish one study, and you are waiting for data analysis, and you trying to start another study, and you recruit another study sites to do the studies. And that's the difference. When you do the one protocol, everything is designed from the very beginning. But you have many stopping points. I'm going to pass over to Ken.
Yeah, and so the idea is the operational aspects are more efficient, setting up contracts with sites and setting up the case report forms and so forth. And for one study is theoretically, more efficient than having, and cheaper than, and faster than having to set up, 2 or 3 studies. So if you have 2 or 3 studies in one study, the operational aspects can be more efficient if it's planned out carefully, and if you have a good project manager like Lei involved with your studies. So yeah, that's one advantage too, is the operational efficiencies too.
Yeah, my question is on the adaptive design. In the phase 1B study, multiple dose escalating, you recruit healthy volunteer, and then you move to the patient, or you recruit patient?
That would be 2 different studies protocol. So basically, you do a phase 1 study, healthy volunteer. What we usually do now is we pick a country, 1 site, and then we run the healthy volunteer really quickly, 6 cohorts to 8 cohorts. And we finish that study in a very short time, few months. And that's one study protocol. You only contract with 1 site. We finish that study, and we have the dose range, and we kind of know that which dose range is safe. And we pick 2, maybe 2 dose range, and do a run-in really quickly. Is to the disease population. That's the adaptive study design start. We separate we won't use the same site, the healthy volunteer, to the disease population. And usually, because the adaptive study design, we increase the study size as well, because it's going to be a huge study. Because one protocol, we need to cover run-in stage. We have stage 1, run-in, 2 cohort, And we have stage 2 of the phase 2 of the study, the stage 2 of the study. Once we finish that, what's the outcome going to look like? And the safety monitor committee, they review the data, allow us to make a decision, pick one dose, and continue the dose, repeat the dose, but pick the disease indications. We enter the POC stage. And then that time, the cycle start, because I will say each cohort, I will run 20 patients, or here it's like 30 patients, but this one is for it's bigger size because this one is for phase 2/3, actually. It's study design, the example I'm picking up. But the similar type of model we use for the early stage, phase 1/phase 2 POC, which usually is a smaller size. And we will do that a cycle. Again, we finish, and it's a double-blinded study. So we will run a cycle of cohort, and we stop. Interim data analysis. The safety monitor study, the committee, they will review the data because we will have a blinded statistician. They will do the calculations, see if we can see the efficacy, and depends on our endpoint decision, and we see the safety. And if it's okay, we haven't seen the efficacy, but we see the safety is okay, they will give us a go. We go to the next cohort, and then we run 20 patients and then we stop. Interim data analysis again. So it's many stop and go. But it's good thing is only one study, it's one study protocol. So the most part, the efficiency of the adaptive study design is stopping from if you have one study protocol, every time you have new study protocols for study initiation, you need to submit your protocol to regulatory agency. And the bigger study, you need to go to different countries for country activations, the regulatory submission, regulatory approval for the the country activation. And then you need a time. That will take about 3 to 4 months if you have a different country, especially EU. They have to go through the health authority. You have to go through the ethic committee. And then that take about 3 to 4 months. You contract with the site, and then you contract with the site. Finish the contract with the site. Each site opening is very expensive. And then you start a site, and then they start enroll the patients. So usually we're talking about the site. If you have each study, the enrollment from the start the contract, you had a protocol to the submission for the regulatory approval to study to activate the site to start the first patient enroll in that site. It's taking about the small study or the early stage is 6 months. The later stage can up to 12 months to have that the first patient in. Not talking about enrollment. So the time losing, if you have 2 study, 2 protocol, you stop here and go. You are talking about in between 2 study, you have 6 months to a year, 12 months of time loss there. And then you recontract with the site, reinitiate the site. That's another initiation fee. When you sign with the site, there's an initiation fee. And then how many patients enroll, how many patients per patient, how much you pay. So you have to know that's why the adaptive study design becomes so attractive. And then also it becomes stopping and go, evaluate the data. So you got to know you cannot get to see the early signal as early as possible. You are not waiting after you run 6 cohort and you finally see the data. So that's kind of something different too. Yeah, good. And again, today we have very, very good discussion in all 3 tracks. And so I think you will like the content we are going to provide it to you. Thank you.